Hi everyone, today I will introduce bacteria to you. This video will cover background, knowledge of transformation, and bacteria transformation's application. Bacteria were observed by Antony van Leeuwenhoek in 1676 with a single-lens microscope of his own design. People of that time saw bacteria as the enemy and studied ways to destroy them. However, with the development of technology, researchers found that they can be used in the biological study as well. They also found that the movement of genetic material between organisms plays a key role in the evolution of bacteria. It is named as horizontal gene transfer in the biology major. HGT includes several ways: transformation, transduction, conjugation, and transfection, which can only be done in the lab. Then I will focus on the transformation in nature and in the lab to expand my introduction. Transformation in nature usually occurs in the same species or related species. Natural transformation has been established in Methylobacterium organophyllum in the Canobacteria synagococcus asp. As it is found in the nature, it is quickly applied in the lab. Bacteria are used as a means to store and replicate plasmids. Gradually, the plasmids are rebuilt by the biologists to find whether the bacteria can replicate the expected gene carried by plasmids. Then, a new question arises. How can we transform plasmid into bacteria, and what kind of bacteria can do this work? After a lot of experiments, biologists found that bacteria such as Streptomyces, Pseudomonas, Acinetobacter, Moaxilla, Neisseria, and Achromobacter can, and the experiments in the lab are interesting and easy to attempt. First of all, in order to make the comparison, we should also add some controlled experiments to test whether it is transformed correctly. The difference is the plasmid. The inserted plasmid should have the ability to resist antibiotics like penicillin, azithromycin, clindamycin, and so on, while the bacteria will die as long as meeting the antibiotics. In this situation, we can distinguish it clearly. Here, I want to add some more information about plasmids. Plasmids are most from bacteria and will not interfere with the DNA in bacteria. It will replicate by itself since it has replication original in the sequences. That's why we should pay more attention to the insertion of the DNA sequences, which can resist the antibiotic since we can't cut the replication original. Secondly, we need a competent cell as a receptor cell. The ways to change bacteria, a single-celled organism, into a competent state are calcium chloride method, TSS method, and super-high method. The most common way to prepare competent cell is to treat logarithmic growing bacteria with preclude calcium chloride. It is to treat rapidly growing bacteria with hyposmatic calcium chloride solution at low temperature to obtain competent cells. At this time, the cell expands into a ball, and the exogenous DNA molecules are easy to form hydroxycalcium phosphate complexes that resist DNA enzymes and adhere to the cell surface at low temperatures. Then we do some heat shock, which can promote cell absorption of foreign DNA molecules, and then this transformed bacteria can resist the antibiotic which it cannot before. Above are the simplified steps for bacteria transformation. Finally, let's take a look at the medical application of bacteria transformation. Natural transformation has been applied as a tool for genetical manipulation since its discovery. In recent years, natural transformation has become a part of recombinant DNA technology and has been applied widely in insertion mutagenesis, cloning of genes into new hosts, and and the structural mapping of chromosomal genes in a variety of bacteria. Natural transformation has also been applied to taxonomy. The application has already had promising results. For example, the genetic relatedness of 109 isolates of a chromobacter and their unrelatedness to type strands of molecular and acetobacter were demonstrated by transformation. There is a lot of information from biologists' research I haven't covered. If you are interested, I have put some website and book references I have used in the comment area. You can look for them by yourself. Thank you.